All right. Well, thank you very much for inviting me and for this opportunity to share with you some thoughts about epilepsy genetics and particularly the role that the EEG can play in helping us to better understand epilepsy and ultimately better treat it. So our subject today, and I won't, I'll be relatively brief so that we have time for questions. Um, but our subject today is pretty extensive. I mean, when you stop to consider it, there are 100 stars in the galaxy, and that's how many neurons there are in our brains. So the neurons are the cells that do the thinking in our brains, and about 100 billion of them inside of each of our brains. It gets even more mind-blowing when you think about connections. There are an estimated 100 trillion connections in our brain. This is a image showing diffusion tensor imaging, just showing some of the connections of the brain that we can see grossly between major areas. But when you consider that on an astronomical term, we have, this blows my mind, we have 50 times more connections in our brains than there are galaxies in the universe. Is that crazy or what? Well, most of the time, it works really well. But when it doesn't, we've known for the past about 150 years that you can get epilepsy. And as John Jackson postulated in 1873, epilepsy is due to a sudden, excessive, and rapid discharge of brain cells. I mean, he just hypothesized this, which was sheer brilliance and turned out to be correct. So how do we conceptualize epilepsy nowadays? This diagram from the International League Against Epilepsy, I think, is very helpful. These are kind of our modern thoughts of how we try to, as clinicians, physicians, nurse practitioners, think about epilepsy. That at the top, we try to do our best job at identifying the seizure type. So what, is the seizure focal, meaning does it come from one region of the brain, or generalized, coming more diffusely from the brain? And then we try to arrive at the epilepsy type. And again, you see these broad categories. Is it a focal type of epilepsy or a generalized? Or do we not know? Is it unknown or maybe a combination? And then ultimately, we try to get to the highest level of precision, which is the epilepsy syndrome. And that was the be all and end all, I would say, in the 1980s, 1990s, even going into 2000, was can you arrive at the epilepsy syndrome? And then genetics, shown on the right-hand portion here in terms of one of the critical causes of epilepsy, burst into our world. And suddenly the availability of next generation sequencing completely transformed our lives within epilepsy and also child neurology. So how do we put these two pieces together? How do we put together the genetics on the right-hand side with the information about epilepsy? And I think the key to this is the EEG. Now, EEG, as you well know, is not new. It was kind of really invented by this man, Hans Berger, who discovered the clinical application of EEG many years ago in the 1930s. And here is his first EEG apparatus, which is remarkable, right? But at the bottom, you see that he recorded brain rhythms. In this case, the recording was done on, in many cases, on his son. And you can see normal brain activity, the so-called alpha rhythm that was illustrated at the bottom, which opened up the door to a transformative way of thinking about epilepsy. So my objective here is to help us link these two things, the older technology of EEG on the left and the newer technology of genetics on the right. And in so doing, I hope to give you a sense of enthusiasm about what is happening in the field and why I think we are poised for like some major breakthroughs within epilepsy. So one way to put it is that we're on this, precipice of a new era. And what's the evidence for that? It's not just in epilepsy, but you may have heard about these remarkable advances for muscle diseases, including spinal muscular atrophy, new enzyme replacement therapies for neurodegenerative diseases like Batten's disease and Pompei's disease, and therapies for mucopolysaccharidoses, bone marrow transplantation, techniques for other neurodegenerative disorders like adrenoleukodystrophy. And then the thing that is so exciting in epilepsy is this very recent genetic specific treatment called ASOs. And we'll touch on this a little bit, and the key to understanding how we can figure out this and diagnose and open the door to these treatments. Okay, so our subject is kind of broad, right? Genetics in epilepsy. And you think about it, there are many different ways that genes could play a role in epilepsy. And it could be a dominant trait, or it could be a recessive, where you have to have a combination of two genes to get that condition. Or it could be many genes kind of coming together to conspire to make a susceptibility. And then you can have not only like the standard gene inheritance patterns coming from the nucleus of our cells, but there's also important genetics from the mitochondria, the powerhouses of cells. We won't touch on that today, but that's an exciting chapter in and of itself. The most important, relevant for us today, that we're going to focus on are the so-called de novo mutations. Now, what does that mean? Each one of us has about 15 new mutations that we are presenting with. And many times, those don't really have any impact on us. But what we're discovering is that for certain forms of epilepsy, particularly harder to treat epilepsies early on in life, this is one of the predominant causes, the so-called de novo mutations. So newly arising mutations that are causing the epilepsy and some brain dysfunction. So some years ago, colleagues of mine and I put together this simple categorization scheme for EEG. And I think it's so straightforward that I've been using it for the past about 7 years in my teaching with the residents and fellows. And I think it's a very easy way to understand epilepsy. And so I'm going to use that as a way to kind of guide us through this genetics. So it comes down to that there's only 5 major categories of EEG changes that you see in all patients with pediatric epilepsy. And you can see the first 3. The importance is that the EEG background, which is the activity at baseline in the EEGs, is normal in the first 3 of these. In the last 2, it's abnormal. And in the epilepsies shown with the encephalopathy and kind of a reddish color, there's diffuse slowing of the background. In the epilepsies associated with a focal structural lesion, there's focal slowing. Okay. Now, this information has been packed into the EEG literature way back when. Our predecessors discovered some really essential things that are present in the EEG that help us to understand the cause of epilepsy. And some of these individuals are the Gibbs. The reason I'm showing them is they lived and worked for many decades in Chicago. So it's kind of a Chicago perspective to look at epilepsy from the standpoint of EEG. Let me show it to you this way. This is all of the epilepsies, and you can break them up very simply into epilepsies that are associated with diffuse spikes on the EEG on the left-hand side, and epilepsies that are associated with focal spikes on the right-hand side. On the top, are all the epilepsies where the EEG background is normal, and on the bottom where the EEG background is slowed. That's it. If you know about the background information, the location of the spikes, you know an awful lot to categorize the type of epilepsy that you're dealing with. So how does that relate to genetics? It's the key to understanding genetics. So the first pattern that we're going to touch on, where the EEG is actually normal, the genetics plays a very, very big role. Those epilepsies are very powerful familial traits that are running through families. They're rare epilepsies. You'll encounter them every now and then, but they're rare. So who do we test? I'm going to cut to the chase. Category 1 sometimes, but most importantly, category 4. So I will get to this a little bit more. Okay, now here is the key point about EEG. And you may or may not see this in your sons or daughters' EEG reports, but I've been encouraging the EEGers when they're reporting results to put it in this way. There's only 2 flavors of spikes that you see in the EEG. One is called stereotyped, where every spike looks like its neighbor, very, very similar, looks exactly the same. That has been known since the 1960s to be inherited throughout families as autosomal dominant, a dominant trait that penetrates 2 families, and majority of people in the family, each successive generation will have that trait. And if you do their EEG, you'll find it. So that's called a stereotyped spike. And what's basically telling us is that there is a single component, one gene, that's driving that EEG pattern. In contrast, the other flavor of spikes are called pleomorphic, where every spike looks different from the other. They're very varied in how they appear. And that occurs in the symptomatic epilepsies. So it's going to become important for us from the genetic standpoint for de novo mutations. So stereotyped tend to be running through families, often actually epilepsies with very good prognoses tend to be, easily treated. Pleomorphic spikes, a little orange light or red light should go off, like, ooh, that could be either structural or symptomatic and might be more challenging, and therefore we need to do further investigations. So the first type of epilepsy, I'm not going to spend a lot of time on because these are rare, but these are epilepsies where the background is normal and there's very few spikes, and they are driven by a gene that's expressed in a dominant fashion through the various generations. It might look like this, for example. If you say that the red is color is an individual who has the gene and has the epilepsy, and you can see with each generation that trait passes through in a dominant fashion. That's autosomal dominant inheritance. Not hard to know when you're dealing with this. The family history is very positive. Lots of first and second degree relatives with epilepsy. What we're finding out is that these epilepsies, you can often figure out that they are due to 1 or 2 different mutations. In the case of nocturnal frontal lobe epilepsy, it's due to a mutation in this gene, the acetylcholine receptor. And I published with co-authors on this. You can also see it due to mutations in a gene called DEPDC5. If you put it together, this list, and again, we're not going to spend time on this because they're relatively rare epilepsies, but the importance is that the EEG is normal and the family history is very positive. And then if you just know what type of epilepsy you're dealing with, when it's presenting, what it looks like, there's usually only 1 or 2 genes to check to see if indeed that is the diagnosis. So not a big issue, not a big public health problem from the standpoint, usually these epilepsies we can control with standard medications. So let's move on and get to the most common forms of epilepsy that I want to spend a little bit more time. This second one we'll call type 2. And this is where you have generalized spikes on the EEG, but with a normal background. And now the family history, about 20 to 30% of the time, you'll hear that there are family members with epilepsy, with this type of epilepsy. A good example of this would be juvenile myoclonic epilepsy, where you have normal development, then early morning jerks and convulsions, and a photoparoxysmal response, where you get an abnormal response with flashing lights. And the importance is that you can get an excellent response to medication, even if you may have to take it for a prolonged. This is an example of so-called polygenic inheritance. So what we mean here is, although these are called the genetic generalized epilepsies, there's only a smaller component of genetics that's contributing to this. It's maybe multiple genes are coming together to make someone susceptible, or it may be other factors are lending a contributory factor. So is it worth checking genes in patients with genetic generalized epilepsies? No. By and large, you are not going to find mutations. So it's a waste of resources to check that. Even though it's called the genetic generalized epilepsy, it's a little bit of a misnomer in that regard. It is not going to be useful to check genetic testing in these conditions. So these are one of the commonest forms of epilepsy that we see, ranging from myoclonic epilepsy in infancy to myoclonic atonic epilepsy, as described by Doza. Childhood absence epilepsy, very common. Epilepsy with eyelid myoclonia, so-called Jevons syndrome. And then the adolescent forms, juvenile absence epilepsy, juvenile myoclonic epilepsy, and epilepsy with GTCs alone. There is a more recently described adult form of adult onset myoclonic epilepsy that may also fit into this category, but there you have it. These are very common generalized epilepsies, easily determined, normal background on EEG, stereotyped generalized spikes, and genetics is not very helpful. Okay, the other super common form of epilepsy that we see all the time in pediatrics are the so-called self-limited epilepsies. Again, normal background, by and large normal children, normal development. EEG shows stereotyped, now instead of generalized, focal spikes. A classic example of this is Rolandic epilepsy, one of the commonest forms of focal epilepsy that we'll see in school-age children, where they have nighttime seizures with convulsions. And here again, there are rare families where genetics are positive. The vast majority of patients with these forms of epilepsy are going to end up with normal genetic studies. So not worth checking in these self-limited epilepsies. Now we get to the most important part of the talk, the type 4 epilepsies. So what are these epilepsies? These are epilepsies where there may be some developmental delay at the start, or it may become apparent like into the epilepsy. The background is slowed for age. The EEG background is slowed for age, and the spikes are pleomorphic, not stereotyped, and often in many different places in the EEG. Within 10, 20 seconds of EEG, you know you're dealing with this kind of an epilepsy with very commonly. And this comes in two flavors. The first, let's call it epileptogenic, where an underlying cause produces the epilepsy and brain dysfunction. Our word for brain dysfunction is encephalopathy. A classic example of this is DRAVET syndrome. Used to be called severe myoclonic epilepsy of infancy, described by this absolutely brilliant woman, Charlotte DRAVET. And this is a clearly recognized syndrome with onset of seizures in the first year, often starting with prolonged febrile convulsions. And then you see a development of myoclonus, focal seizures, sometimes atypical absence. And what we know is that this 90% of children with this kind of presentation have a mutation in a gene of called SCN1A. So this has just been, I mean, this is one of the most important genes in pediatric epilepsy. And this has been explosive since this information came up. And so literally you can see, like, every week there are several articles on Dravet syndrome. And I have to tell you, when I started in the 1980s, it was rare that you heard anything about Dravet syndrome. You know, an occasional report here and there, mostly from Europe. And now you can't go literally a week without hearing several very interesting reports on this condition. Here's just one example of a recently published article showing that it's not the seizures necessarily or the EEG spikes that are causing the behavioral and cognitive issues in Dravet. It's uncoupled from that. It may be directly related to the gene mutation. So what is the gene mutation? The gene mutation involves this sodium channel in the brain that targets certain brain cells. And because it lowers the function of those brain cells, it hits the brain cells that are inhibitory. So because it's hitting brain cells that are inhibitory and driving down their function, it causes hyperexcitation or epilepsy. And so we have gone from knowing nothing about the cause of Dravet to probably the most detailed information about what causes the epilepsy in these patients in the past 20 years. And this has led to so many exciting things. This paper was just published. I'm just pulling it out somewhat randomly, but this is a serotonergic medication, fenfluramine. And this was a nice open label study by a Californian investigator, Joe Sullivan, and others. And look at the startling results that they're reporting in open label. So once you know what is causing the epilepsy, and you can get pinpoint precision on the categorization, then you can start to do very selective treatments like this. And look at the right-hand side. The right-hand side is the percent reduction in convulsive seizure frequency and the percentage of patients. So 78% of patients are getting some impact, but 64% are getting their convulsive seizures reduced in half. 41 have a 3/4 reduction, and some even 100% reduction. Looks like the impact is greater in the younger children. And this goes along with an experiment that was done looking in an animal model to see what would happen if you put some of these modulators in an animal model. Could it change the behavior of the nerve cells? And the answer is yes. It looks like it could. It could potentially change the long-term outcome based on the basic science. So very exciting to think that medications that are at our disposal now that are coming up may have potentially long-acting impact. So it doesn't stop there. But just another recent publication published this year, 2020, looking at what are all the options for us, what's coming out, what's already market approved. Well, stirpental is there, you see cannabidiol there, fenfluramine, which we just talked about. But then look at some of these other in products that are in phase 2 or phase 1 or just in pilot stages. And these are novel kind of compounds with different mechanisms of action that people are exploring, which is really exciting. But check this out. It goes even one step further to antisense oligonucleotides. So this is the so-called ASOs that we talked about. And ASOs in this animal model, in a mouse model, have been shown to reduce seizures and lower the incidence of SUDEP in mouse models of Dravet syndrome. So the concept here is that you can introduce these very small compounds, and basically they change the genetic expression. So they give different instructions to the cells so that they hope to correct for the deficiency in the protein expression, which is pretty darn remarkable. In the case of not epilepsy right now, but in the case of muscle diseases like spinal muscular atrophy, there's even an AVI vector. So in other words, you take a portion of a gene, a plasmid, you attach it to a virus, you inject it into an animal or a patient, it goes into the cell and places itself there and corrects for the dysfunction in the cell. So it's not science fiction, that's happening right now in muscle disease. And so we are on the verge of, a terribly exciting time in epilepsy, thanks to this next generation sequencing capabilities. The other type of epilepsy associated with diffuse brain dysfunction, I'm going to call epileptic. And here, this is where there's something about the epilepsy that worsens the encephalopathy. And this, because it grows over time, is where we say time is of the essence. Years ago, Professor Odahara considered it a spectrum of conditions presenting in the newborn period and extending out into West syndrome, and then ultimately Lennox-Gastaut syndrome. What's common about all of these, and I keep teaching the residents and fellows this, is that the EEG isn't always on. It sometimes pauses and is discontinuous. And the important thing that I tell the residents and fellows and medical students is that if you see this, assume that time is of the essence to get control of that child's epilepsy. This was the classic example of this, EEG-wise, is hypsarhythmia, as described by the Gibbs that I showed you earlier in the 1950s. And this, of course, is the EEG pattern that's associated with West syndrome and infantile spasms. So how did we used to approach these things? Well, we used to approach them, and we still do, at the bedside, listening to you, the parents, the families, tell us about the seizures, the details of them, so-called semiology, and then an examination of the child. And then we used to follow with neuroimaging, which we still do. And then if that didn't give us a clue, we would go to metabolic evaluations. The metabolic evaluations used to be very lengthy. Well, it turns out Dr. Anberg, a colleague of mine, asked me to review a large data set from the Pediatric Epilepsy Research Consortium. And Dr. Berg was interested to know what's more useful, metabolic tests or genetic tests? And the answer was genetic testing, far and away. The metabolic testing that came back positive was small, only 14 in this case series of literally 784 children. But when I went through this in detail and looked at these cases, what I realized is that most of them were already suspected and already the clinicians were clued in based on either newborn screening or MRI findings or simple blood test results. So the conclusion from this is that this previous flow that we had where we would, say, okay, history negative, physical exam negative, EEG doesn't tell us what it is, imaging doesn't tell us, let's go to metabolic investigation. We've changed, and what's replaced it is genetic evaluation. So this article was another game changer for me. This was out of the folks in Kansas City, and this was a proof of principle. They basically did whole genome sequencing for genetic diseases in newborn nursery. And what they did was they came up with a very kind of logical, sequential approach to these patients. And they obtained the blood sample at time 0, they sequenced, they looked at variants, and then they did analysis of the variants. And look at this, in 50 hours, they had a definitive diagnosis for these patients. So that is amazing. And what we've seen with the next generation sequencing is the turnaround times dropping more and more and more, to the point where we can get information back far more quickly than we ever used to get from our previous metabolic tests. So what's happened is that technology has driven our understanding of the causes of epilepsy from, the 1800s, where it was just history and physical, to chromosomal analysis. The guy above the chromosomal analysis is Professor Down, person who described Down syndrome. And then we went to things like mass spectrometry, which I spent a lot of time training on when I was in college, to cell sorting, which we started to use when I was in medical school, to advanced MRI imaging, to next generation sequencing. And then with the advent of supercomputers, the possibility of doing entire exome sequencing and analyzing that data. So we moved from simple causes of epilepsy to very specific genetic causes, including channel problems. And now we're on the verge of a whole new era of epigenetics, which is really exciting. Simultaneously, EEG went from those early descriptions and the technology that I showed you to modern techniques, let's call it electrical neuroimaging, so that EEG is keeping pace with the same changes. What we need to do now is link the two together. We need to link the dynamic, time-dependent information from EEG with the genetics. So a number of us are very interested in forming biorepositories where we put together information derived from the genetics with the information from EEG, so that we learn how do these underlying gene changes influence the EEG, and then therefore how can we use the EEG to predict outcome and to see if we're getting good responses to treatments. So what's our modern flow now? If we have an early onset epilepsy, and we have one of these EEG patterns that I was describing to you, the type 4, and there's no explanation on our history and physical or the MRI, and some simple metabolic tests are negative, just very simple, screening, ammonia, glucose, lactate pyruvate, then the most important thing is to move to genetic testing. Thank goodness there are these initiatives. So in the Behind the Seizure, there is the capability of testing everyone 7 and under who develops epilepsy at no charge to the patient with a fast turnaround time. So this has opened up the opportunity to get genetic testing on everybody that we feel could potentially benefit. So what's on the horizon? These ASOs, and they come in a variety of different flavors for genetic epilepsies. Outside of this conversation, we have imaging advances and surgical advances. There is another whole fascinating area in epilepsy of the role of the microbiome and how that's influencing inflammation. And then that leads to a greater study of what's happening with the entire immune system, both innate and acquired. So there's a lot of interesting things going on in pediatric epilepsy, and genetics is one of the key, most exciting elements. So what I hope that I've shown is that EEG findings can help us understand the causes of epilepsy and particularly guide us as to who to evaluate with genetic studies. That's that category 4, early onset, no known cause, EEG that shows slowing and multifocal spikes, unremarkable imaging, really good candidate for epilepsy genetic studies. I've been thinking for some time that it's time for us to change our attitude. Maybe we've been too passive, and maybe we've been too guarded, and maybe we need to allow ourselves to think about markedly changing the landscape in pediatric epilepsy. And maybe we need to be bolder about what our goals are. And I think that with all of these things that are coming up and at our disposal, we need to start allowing ourselves to think that there may be a time, let's hope in this decade, where we could eliminate refractory epilepsy. Now, it sounds kind of crazy, but is it possible? And I'm not going to show you here, but just mathematically, if we got reasonable success from any one of a number of these contributory factors, I think, yes, it's mathematically possible. So let's hope there's a very bright future ahead. And I think we need to change our consciousness from being passive to being proactive when it comes to pediatric epilepsy. With that, I'm going to pause just to introduce my team. This was pre-pandemic when we could still do things like this and get together and be close to one another. But this is the team from University of Chicago. And I'm a little bit ahead of time, but that's okay. I'm going to pause here for questions, comments, and be very happy to entertain any questions that you might have.
Okay, we have a couple of questions people have written in. So the first question is, as far as the type 2, I'm sorry, I lost my chat. Give me a second here. As far as the type 2, they're asking, can it skip multiple generations?
Yes, it can. The type 2, the genetic generalized epilepsies can skip generations. So about 30% of people, of families will have another family member. So you may not find family members in all of the genetic generalized epilepsies, but about 30% of the time you will. And yes, good question. It can skip generations.
Okay. I just saw a question here, and I had to grin because every time genetic epilepsy comes up in a discussion, somebody throws out the name of yet another genetic epilepsy or another offshoot. So we have a question regarding Unverricht-Lundborg disease.
Yes, very good question. Unverricht-Lundborg is a form of progressive myoclonus epilepsy. And this is a rare genetic condition, but very important cause. So what does this look like? It can initially look like a GGE, like a regular genetic generalized epilepsy. But what's different is that you may have after it looks like, well, it's just convulsions and maybe myoclonus, then you may have difficulty walking, so-called ataxia, and some cognitive issues. It is notoriously enhanced by activity. So it may trigger some of the seizures. It's something that's being along with Lafore disease, its cousin. and it's an important form of epilepsy that fits into that category 4.
Okay, I have one other question here. My son has Lennox-Gastaut syndrome, and it's very hard to treat. What is it? Is only 2? Do you have? Oh, he's only 2. Do you have any advice for somebody who's has a son with LGS?
Yes, I do have advice. It's early to have Lennox-Gastaut syndrome. Typically, Lennox-Gastaut syndrome properly will start up a little bit later in childhood. So therefore, I would, maybe say, just have a conversation with your neurologist. Say, well, is that exactly the diagnosis? Or could it be something else because it's a little early, and it may be worth doing more investigations to try to characterize what kind of epilepsy that is. It's just statistically early, and so it's likely that it's not exactly Lennox-Gastaut given the age of your son, and it might be worth doing some more thinking about what could be the potential cause.
Okay, the questions keep coming in. Just give me a second here to scroll down. Is there a relationship between Ehlers-Danlos, hypermobility, and generalized epilepsy?
Excellent question. Not to my knowledge, but I wouldn't take that as gospel, because every day or week there are new things that are coming up. One of the nice things about our modern era is that we can go into really good resources like Google Scholar, for example, or PubMed, and rapidly search for those kinds of potential rare associations. So I don't know that offhand, but again, don't take that as gospel. It might be worth doing a little bit more searching to see if there's any known association.
Okay, another specific marker we were asked about, genetic mutation GABRB2.
Mm-hmm. Yes, a potential cause of epilepsy. And thank you for that question. There's a really important point embedded in here. So when we get the results back.
Jenny, I'm having a hard time with that. I'm sorry, Doc.
Oh, that's okay. When we get the results back of genetic testing, sometimes we get an answer that we're expecting. Like, it makes perfectly good sense. Like the mutation matches what kinds of epilepsy we know. Sometimes we get back an answer that is not 100% clear, and that's often called a VUS. Stands for a variant of uncertain significance. And so the importance here is to, number one, see, does it match what the epilepsy looks like? And then number two, has it been reported as a mutation in that disease, in that same condition? If it hasn't, it still could be a valuable answer. So how do we figure it out? Well, we have a number of different tricks. One is to study the parents, to see if the parents have it. The argument here is that if we believe it's a non-familial epilepsy, then the parents shouldn't have it. So that's called examining the trio, checking both parents. Another thing is to look to see, is it an important area of the gene, so-called conserved area? Does it change the chemical structure? Does it result in an amino acid change? And if so, do models predict that it's going to be a major change that could disrupt function? So we have a number of tricks to look at, but it's a really important point. Yes, it could be an answer to your child's epilepsy, but you need to see if it correlates, and you need to talk with your doctor about some more details about the posted mutation. Really good question.
And this question I love, that's asked every year at the expo, and honestly, every year the answer changes because of the new research that's coming out. How many epilepsy-causing genetic mutations have been found, and how many ASOs have been discovered to treat those specific mutations? And then they're also following up with the question: do you believe that there will be one day a treatment available for each of these mutations?
I am very excited about the opportunity. So yes, I do think we are going to see treatments available. Now, I can't say for all, but there are about 100 genes that I think play an important role in pediatric epilepsy. And right now, I know of about a half a dozen specific conditions that have either specific like enzymes or genetic treatments that are under evaluation. We can't, disclose them. They're under research right now. So it's really exciting. And what's happened, and this is where you all play a really important role, is I've seen this story unfold a number of times. So a gene is discovered, a parent group forms, the parent group starts to raise interest in the condition. Maybe they even raise some money. Maybe they start supporting a little bit of research that's going on. Next thing you know, people in basic science are creating animal models with that. Next thing you know, there are these medium-sized companies that say, we have an ASO that we think hits that target. And they contact the basic scientists and they run it through the animal models, and you're off and running. So that's what's happening. And families, parents are playing, can play and are playing a really critical role in this whole new exciting phase. So now you say, like, why would a company be interested in just a relatively small number of patients? And the answer is because it's a foot in the door. It's a means of saying this platform can work. And if it can work for these rare conditions, maybe it can work for more common conditions. So there's plenty of interest out there in these medium-sized companies that are interested in exploring this. So it's like a perfect combination of factors coming together to make us optimistic for the future.
Nice. Okay, this is another specific question. This writer is asking, she's saying she has two daughters with ADN FLE, and they're currently doing a trial with nicotine, and she's asking if you have any information on that trial.
Don't have the specific trial. It is something that had been reported some years ago by Australian colleagues. And I myself have tried it in some of my patients with ADFNLE with mixed results. So it's logical because the mutation involves the nicotinic acetylcholine receptor. But after I tried that with my patients, I contacted one of the physicians who wrote the original report from Australia, and he himself said, not everybody seems to be responding. So not to put cool water on it, but just maybe to balance expectations. It's attractive. It has been tried, some mixed success. So that's exciting that a more formal trial would be undertaken. Maybe we can uncover a subpopulation that is more responsive. Got it.
Okay. This is another great question that I want to hear the answer to. I think everybody does. This writer's daughter had an epilepsy gene panel done back in 2016. It came back with the diagnosis of ULD, but now that we have all these advances and genetic testing is much greater than it was in 2016, should we retest?
Maybe not if it came up with the diagnosis of Unverricht-Lundborg disease, which I'm assuming that's the ULD abbreviation. Maybe you have the answer. But I mentioned, I know that in a cousin, Lafore disease, that there's a lot of research going on by Berger and colleagues, for example, in Texas. So there is definite excitement within that field of progressive myoclonus epilepsies. So I would stay tuned, and the good thing is you know exactly what literature to search, and then who might be doing, you know, groundbreaking work in that particular area. Let's say it wasn't. Let's say if the child hadn't come up with a known cause, and say it was 4 or 5 years ago, is it worth retesting? Yes, I think so. I had this experience very recently where a panel was run some years ago, more like 7, and then we retested, and it turned out that we picked up the new gene that was the culprit. So we are learning more about what are the highest payoffs to include in these panels. So sometimes it may be worth considering retesting. I went through and I made a list of all the genes that were in the previous panel and the new one, and I saw quite a number of differences. So it's good to stay on top of those things and keep an open mind about retesting if you got negative results in the past.
Wow, we're getting great questions, Dr. Nordli. I'm so happy that they all came in for you, because I know they're getting great answers. Is there a genetic component to focal cortical dysplasia?
Yes, there is, but it's not inherited, you know, like passing from you to the child. We think that what's going on is that inside of the brain, when the baby is developing, that there is a mutation inside some of the progenitor cells, and that in one region, that genetic mutation produces a change in the cells, probably within a pathway most of the time called the mTOR pathway. So it's not at risk for other people in the family whatsoever. We think it's typically just within that child, but it opens the door. What more and more people are thinking is, ooh, if that's the cause, maybe we'll come up with newer treatments that target that pathway. So more to follow. This is a rapidly evolving situation, and where people are now using these same tools for the pathologic specimens that we obtained from epilepsy surgery to uncover this. So really good question. And yes, that is an area of advancement as well.
This next question, after reading it, I almost want to try to help this family get out to Chicago to visit you, because it looks like they might need more of a visit than a question. My son has the chromosome 14q duplication and the 21 DUOLPOHE, and he was diagnosed with IS, infantile spasms. He's on 4 different meds, not under control.
2 things. Thank you for that flattering comment, but there are excellent pediatric epileptologists all throughout the country. I can honestly attest to that there's and they're scattered around and with big levels of experience in all different parts of the country. So I would say very often, thankfully, you don't have to travel far to find out, to get really expert opinions. Without getting into detail, one thing that we've discovered is that in general, and this is a broad statement, if children have early onset epilepsies due to genetic causes, that they can respond many times very well to dietary therapy. So in patients like you're describing that maybe 4 drugs are failing them, very often we will move them towards dietary treatment, including full ketogenic diet. So I don't know if that's been tried or a consideration, but let me throw that out there. And then there are a lot of really good pediatric epileptologists, thankfully, in the United States. So and by and large, we tend to talk a lot to one another. So the advances that somebody hears about can often go through the network pretty quickly. So I hope you can find some good help, and hopefully those comments help.
Okay, I think we just got a couple more questions here for you. Um, we have a question. Is there a link between SIDS and epilepsy? My son is a SIDS survivor and developed epilepsy at 27. His grandfather's twin died of SIDS.
There may be another hot area of research. I was part of a NIH lab without walls that looked into SUDEP, and one of my co-investigators is George Richardson from Iowa. He's an adult neurologist, and he studied SIDS. And so some of the mechanisms with the serotonergic neurons may be similar in SIDS and SUDEP. And recently, not too long ago, the group at Harvard published that there may be children who were diagnosed as having SIDS, but actually had some kind of malformation in the hippocampus, raising the question as to whether, did they not really have SIDS, but did they have epilepsy that was undetected? So a really good question. There's another area of investigation that I stumbled upon when I came to University of Chicago. There may be a connection between the two. Too early to tell, but that's one of the pilot studies that we have ongoing. So yes, there may be some connections, but it may be the minority of cases. It may not be very common that you'll see SIDS and then epilepsy in the same family.
Oh, okay. Let me see if I have anything else here. Man, this is a bit specific. I shouldn't read that question. I think unless anybody has any more questions, I think Dr. Nordli has pretty much addressed them all. So nice to have you, Doc. I really appreciate you making the time, and we miss you in California, sir.
Miss you guys too, and it's a great place, and you guys do great work. Keep it going, and thank you for including me.
Thank you, sir. We'll see you soon. Take care.